Welcome to the PubMed Daily Review for April 7th, 2026. Today we scanned 219 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5. Alcohol use, aging, and brain. This trend examines how alcohol impacts brain health across the lifespan, considering genetic, medication, and neurobiological factors. This cluster covers 33 papers. Representative work includes morbidity and mortality in alcohol use disorder, the role of comorbid substance use disorder, age, sex, and the A1 allele of the TAC1A, RS1800497, polymorphism in the ANKK1 gene in an 18-year follow-up, alcohol medication interactions among older people, a narrative review on age-specific evidence, and economic effectiveness of pharmacogenomics-guided prescribing for psychiatric disorders, a systematic review and meta-analysis. Trend 2 of 5, Materials Science and Biology. This trend explores the intersection of material properties and biological systems for applications in health, energy, and agriculture. This cluster covers 66 papers. Representative work includes accelerated mechanochemical depolymerization of polystyrene due to formation of a cohesive state, silicon-rich amorphous SiC-X for the lithium-ion batteries. How does strong carbon doping affect the lithiation behavior and electrochemical performance? And hesperidin alleviates systemic inflammation and oxidative stress by remodeling adipose tissue lipid metabolism in paraparturient dairy cows. Trend 3 of 5, Materials, Biology, and AI. This trend explores AI's role in understanding and manipulating biological systems and advanced materials. This cluster covers 43 papers. Representative work includes evaluation of structured and C-blend refuges for corn earworm, Lepidoptera noctuity, using emergence cages and screen-bagged ear methods, bis, oxalato, borate salts as safe and fluorine-free alternatives to conventional supporting electrolytes for organic electrosynthesis, and tape casting of LLZO ceramic separators. An overview of challenges, optimization strategies, and paths to industrial implementation. Trend 4 of 5. Clinical interventions in health. This trend examines clinical interventions and their impact on various health conditions and patient outcomes. This cluster covers 51 papers. Representative work includes clinical effectiveness of monofilament fiber pads for bacterial and biofilm removal in pressure injuries, a retrospective cohort study, intraoperative hemodynamics, and anesthetic implications in superobese parturients undergoing cesarean delivery, a retrospective cohort analysis, and effects of sensory constraints on sensorimotor functions in people with chronic ankle instability, a systematic review and meta-analysis. Trend 5 of 5, Musculoskeletal and Evolutionary Biology. This trend explores the interplay between biological structures, their evolutionary adaptations, and the impact of disease and aging. This cluster covers 26 papers. Representative work includes aging-related structural and cellular alterations in the mouse muscle tendon junction, brachiocervical inflammatory myopathy, multilevel clinical histopathological and multi-omic analyses of a syndrome variably associated with systemic sclerosis, and empirical validation of the nearly neutral theory at divergence and population genomic scales using 144 placental mammal genomes. Up next, our first paper out of 5. In vivo CRISPR/Cas9 screening reveals that UB2L3 modulates autophagic flux through TSC2 ubiquitination and potentiates PD1 blockade in triple-negative breast cancer by Jian Su and Huaidong Cheng from Department of Surgical Oncology, the first affiliated hospital of Bengbu Medical University, Bengbu, Anhui, China. Let's break down its key contributions. Triple-negative breast cancer, or TNBC, is a particularly aggressive form of breast cancer that's challenging to treat. Unlike other subtypes, it lacks specific targets like estrogen or HER2 receptors, leaving fewer options for effective therapy. Researchers are exploring new ways to combat this disease, looking at fundamental cellular processes like autophagy and how tumors interact with the immune system. To uncover new therapeutic avenues, the researchers employed an in vivo CRISPR/Cas9 screening approach using the aggressive 4T1 triple-negative breast cancer cell line in mice. This powerful technique allowed them to systematically knock out genes and observe the effects on tumor growth and response to therapy. They then used a variety of molecular and cellular techniques to pinpoint the exact mechanisms involved, focusing on how specific genes influence autophagy and the tumor's interaction with the immune system. The study revealed that UB2L3 plays a critical role in TNBC progression. Knocking down UB2L3 significantly restricted tumor growth and, importantly, increased the infiltration of CD8+ T lymphocytes. This enhanced immune cell presence translated into a heightened sensitivity to anti-PD-1 therapy. The mechanism involves UB2L3 interacting with SMUF2 to control the ubiquitination of TSE2, ultimately impacting cellular processes and the tumor microenvironment. These findings highlight UB2L3 as a potential new target for treating triple-negative breast cancer, offering hope for more effective therapies. By understanding how UB2L3 influences autophagy and the immune response, we can potentially improve the effectiveness of existing treatments like anti-PD-1 therapy. This research opens exciting avenues for developing novel treatment strategies, possibly in combination with immunotherapy, for this challenging disease. Up next, our second paper out of 5. Clonotype-Resolved single-cell multi-omics unlocks the profile of tumor-infiltrating CD39+CD8 T cells and enables adoptive cell therapy for solid tumor by Zihan Zhao and Rong Yang from Department of Urology, Nanjing Drum Tower Hospital, Affiliated Hospital of Medical School, Nanjing University, Nanjing 210008, China. Let's break down its key contributions. Immune checkpoint inhibitors have revolutionized cancer treatment by boosting anti-tumor T cells. However, response rates are still not ideal, partly because we don't have enough functional tumor-reactive T cells. Adoptive T cell therapy, like using tumor-infiltrating lymphocytes, TILs, is a promising approach. But finding the truly tumor-reactive T cells among the many present in a tumor is a major hurdle. The role of CD39, an enzyme involved in adenosine metabolism, in these tumor-reactive T cells is debated, making it a key area to investigate. To tackle this question, the researchers used a multi-pronged approach. They developed a mouse model of bladder cancer and also analyzed samples from human bladder cancer patients. Using advanced techniques like single-cell RNA and TCR sequencing, they could deeply profile individual T cells. They then tested the therapeutic potential of CD39 plus CD8 plus T cells by transferring them into tumor-bearing mice and assessing their killing ability in lab experiments. This allowed them to pinpoint the characteristics of these specific T cells and their anti-tumor functions. The results were quite compelling. The study found that CD39 plus CD8 plus T cells displayed clear signs of being active and ready to fight tumors, showing features of proliferation and cytotoxicity. When these CD39 plus CD8 plus T cells were transferred into mice with tumors, they significantly suppressed tumor growth, demonstrating their therapeutic power. Furthermore, T cells engineered with the specific receptors from these CD39 plus CD8 plus cells were highly effective at killing tumor cells in vitro. Importantly, in human patients, a higher presence of these CD39 plus CD8 plus T cells was linked to earlier stages of cancer and better survival outcomes. So what does this all mean for the clinic? This study strongly suggests that CD39 plus CD8 plus T cells are not immunosuppressive, but rather represent a key population of tumor-reactive cells that can be harnessed for therapy. CD39 emerges as a valuable biomarker to help us find and isolate these potent anti-tumor T cells. While these findings are exciting, further work is needed to refine how we select and expand these cells for optimal use in adoptive cell therapy strategies for solid tumors. Up next, our third paper out of 5. FOXO1 mutations co-opt nascent transcription factor networks in partnership with androgen receptor to enhance prostate tumorigenicity by Eric M. Ladewig and Charles L. Sawyers from Computational and Systems Biology Program, Memorial Sloan Kettering Cancer Center, New York 10065, USA. Let's break down its key contributions. Prostate cancer is a complex disease, and genetic mutations play a significant role in its development. One such critical gene is FOXO1, a pioneer transcription factor frequently mutated in about 10 to 40% of prostate cancers. These mutations are known to broadly alter how DNA is accessed, potentially driving tumor growth. This study aims to delve deeper into the functional consequences of these FOXO1 alterations. To investigate the impact of FOXA1 mutations, the researchers analyzed a large cohort of 874 prostate tumors. They then used mouse prostate organoids to model the effects of specific FOXA1 mutations, including common missense, changes, and truncations. By combining multi-omic profiling techniques like single-nucleus multiome, RNA-seq, ATAC-seq, and CHIP-seq, they examined the resulting changes in chromatin and gene expression. These findings were further validated in vivo, in mouse models of prostate cancer. The study revealed that different FOXA1 mutations lead to distinct cellular changes. Indel mutants promoted a more basal-like state, while C-terminal truncations, missense mutations, and even elevated wild-type FOXA1 drove secretory L1, like luminal fates. This L1-like specification was found to involve a novel hybrid androgen receptor/FOXA1 motif and cooperation with another factor, PAU2F1. Importantly, in vivo experiments showed that these mutations, when combined with other genetic losses, shifted tumor histology towards these secretory luminal phenotypes, highlighting their role in enhancing tumorigenicity. This research significantly advances our understanding of FOXA1's role in prostate cancer. It demonstrates that FOXA1 mutations are not just passengers but active drivers that reshape tumor cell identity. The discovery of novel transcription factor partnerships, like the FOXA1-AR PAU2F1 axis, opens exciting avenues for targeted therapies. Future studies should focus on how these specific mutation-driven pathways can be exploited for better patient outcomes. Up next, our fourth paper out of 5, Ubiquitin-conjugating enzymes in cancer by Jiang Yao and Xiaoqing Guan from College of Pharmaceutical Sciences, Zhejiang University of Technology, Hangzhou, Zhejiang, 310014 China. Let's break down its key contributions. Cancer is a complex disease driven by intricate signaling networks. A key player in these networks is the ubiquitin system, particularly ubiquitin-conjugating enzymes, or E2S. These enzymes are emerging as critical regulators of oncogenic signaling, influencing everything from cell proliferation to immune evasion. Despite their central role, E2S are currently underexploited as therapeutic targets. This review aims to systematically map E2-mediated signaling in cancer and propose strategies for E2 targeted interventions. This paper is a comprehensive review, not a primary research study. The authors systematically analyzed existing literature to understand how ubiquitin-conjugating enzymes, or E2S, contribute to cancer development. They mapped the diverse signaling mechanisms orchestrated by E2S across various cancer types. Furthermore, they inventoried current small molecule inhibitors and other agents that target E2S or their regulatory elements, summarizing their efficacy. The review highlights that E2S are central to driving key cancer hallmarks, including proliferation, immune evasion, and metastasis. They play crucial roles in regulating cell cycle progression by influencing cyclins and tumor suppressors. While E2 mutations are relatively rare, they can be pivotal drivers of cancer. Importantly, the study underscores that E2S are druggable targets, presenting significant therapeutic potential. This review emphasizes that E2S represent a promising new class of therapeutic targets in oncology, offering a way to tackle cancer hallmarks that are often resistant to current treatments. The clinical significance lies in their potential for targeted intervention. However, more research is needed to fully understand the complex roles of specific E2S and their mutations in cancer, which will be essential for developing effective E2-directed therapies. Up next, our fifth paper out of 5. The Physiology, Pathophysiology, and Clinical Implications of Fluid Resuscitation in Critically Ill Patients by Akram M. Iraqi and Mostafa Alamrozi from Department of Emergency Medicine, Freeman Health System, Joplin, USA. Let's break down its key contributions. Fluid resuscitation is a cornerstone of critical care, aiming to restore balance in critically ill patients. But how do different fluids affect the body, and what are the best strategies for their use? This review delves into the complex physiology of fluid administration, exploring its impact on acid-base balance, perfusion, and hemodynamics. We'll also examine current evidence and guidelines for fluid use in common critical conditions. This paper is a comprehensive review, critically analyzing existing clinical trials and observational studies. It focuses on critically ill patients across various conditions, comparing different fluid types like crystalloids and colloids. The review examines their impact on physiological parameters, patient outcomes such as mortality and organ support, and overall safety profiles. The review found no significant advantage of colloids over crystalloids in most critically ill patients, with trials like SAFE and CRISTAL showing similar outcomes. However, albumin showed potential harm in trauma and perioperative settings, linked to increased mortality and complications. Furthermore, different albumin formulations have varying effects on acid-base balance, with isooncotic albumin potentially causing acidosis. So what's the key takeaway? Crystalloids are generally the go-to fluid for resuscitation in the ICU. We should be cautious with albumin, especially in trauma and surgical patients, as it may not offer benefits and could even cause harm. Future research should continue to refine our understanding of fluid therapy to optimize patient outcomes.